We actually designed what we thought was an improved drain, which reduced the amount of aqueous and going into the tissues and prevented its pressure from rising above 12 millimeters of mercury. And we thought, you know, this was great, and we did it. This was in just before I left South Africa. And it proved to be a complete disaster. It produced incredibly fibrosed blebs, and we couldn't understand why. You know, the aqueous caused fibrosis. But when you prevented its pressure from rising, it caused, and less went into the tissues, it caused more fibrosis. Now, that was in 1973. So we kept the slides. We studied them, we scratched our heads. Well, there was no clear explanation. And we tried a different tack, and in order to prevent undue drainage, undue rapid drainage when putting implants into very badly damaged eyes, we developed a technique of tying an absorbable suture around the tube. And we found that preventing the aqueous from draining for a while produced a thin avascular layer of connective tissue around the bleb, a very thin layer. And then when the suture dissolved, aqueous went at a high pressure into the tissues, and all of it went into the tissues. That one produced much thinner fibrous tissue. And we didn't really know why and what was happening. So I collected slides, or histological specimens from different types of glaucoma, different severities, and tried to sort of work out what was happening. And in 1990, we developed an implant with a pressure ridge to prevent the aqueous running out too fast. And that worked. I mean, it worked clinically. It was an improvement. And then, after about 5 years, and we used it particularly for neovascular glaucoma, some of these patients died, and I got their eyes. And I noticed that where the aqueous had drained initially, it was particularly, there was a lot of fibrosis, and much less later. And then, with Dr. Alec Dempster, who's a pathologist here, who's worked with me all these years, we noticed that the process by which the fibrous tissue was laid down for about 3 or 4 weeks, then the aqueous seemed to cause the connective tissue to rot and the cells to die. And we identified apoptosis as occurring in this fibrous layer. And then more recently, a lot more has been known about apoptosis. And we realized, in fact, that if the aqueous runs into connective tissue that is less than, if the cells are less than 50 microns, which is not far, 50,000th of a millimeter, from a capillary that's got blood in it, which is all your cells should be, they will respond to the aqueous, which has got a little oxygen and low protein, by being stimulated, activated, migrating towards the flood, and lay down layers of collagen, and they will build a barrier. However, once they've built the barrier thick enough to push the pressure up to above the pressure in the capillary, it'll collapse the capillary, and it finds itself, these cells find themselves in a state of decreased oxygen. They then decide that they then have a different response, a very primitive response. They no longer secrete collagen. They secrete a primitive precursor, or sort of precursor called oxytolin, and they also secrete thrombospondin, which is a slime related to collagen. But at the same time, they are activated, they digest themselves, releasing enzymes which break down collagen and digest themselves into soup, which other cells live on, but mixed in with the soup are death messengers. So the dying apoptosing cell digests itself into soup, literally soup, protein soup for other cells to live on, and mixed in with the soup is a poison pill known as Fas ligand, tiny little particles. And this process spreads through the bleb capsule and makes it permeable and suppresses the inflammation. Now this has recently been recognized as occurring in quite a lot of tissues in the body as being very important. So now that we know what happens, we redesigned the Mark III implant to concentrate the aqueous in a small area, a small primary bleb area, and try and keep it there so that you had a small area of intense inflammation. The pressure then needs to rise and to collapse the capillaries, needs to rise above 15 to 25 millimeters of mercury, pressure in a capillary. Then the apoptotic process starts, the inflammation is switched off, and a cell cycle is established, which makes the bleb permeable. But the pressure then lifts the tissue up off the primary bleb into the secondary one, and that aqueous has been seeded by the death messengers from the cells that have sacrificed themselves to save the glaucoma drainage operation. It's not altruistic behavior on the part of cells, it might be, but I think it's forced. And we've actually got a magnificent histological specimens actually showing this. And this is what we introduced into the Mark III implant. And it's working out, because we are able, with this relatively small implant area, 175 square millimeters, to get actually somewhat better control of intraocular pressure than we've been getting in the past with the two-plate large implant. That's 140, about 275, 280 square millimeters.